Very good afternoon from India, of course, to all of you, and then good evening to our honored speaker today, Dr. J Higgins, because it's almost 7:30 now there in Canberra. So welcome you all to our webinar series towards outcome-based research, biotech research and education roadmap, which we started on 5th of June, and already 2 web seminars we have done, and this is the 3rd one, which will be delivered by a world famous genetic engineer, Dr. J Higgins. And he'll be talking on development of an insect resistant cowpea for smallholder farmers. I welcome you, TJ, to this web seminar. Of course, the people who are working in the area of legume genetic engineer, they know TJ very well. So TJ doesn't need any formal introduction to them, but there are many other participants. So I'd like to very briefly introduce Dr. Higgins to other participants. So Dr. Higgins is currently working as honorary fellow in CSIRO Agriculture and Food, Canberra, Australia. I'm happy to announce here that he is the recipient of Order of Australia Award, which is equivalent to Padma Vibhushan in India. And he is a fellow of Australian Academy of Technological Science and Engineering, and he is also Vice President of Australian Academy of Science. His major contribution is he has been working for nutritional improvement as well as insect resistance in grain legumes. And he has been collaborating with India for insect resistant chickpea with our group, and also with Punjab Agricultural University and University of Agricultural Science, Dharwad, and then IIPR Kanpur through Assam Agricultural University, Jorhat. He has been serving as chairman of our scientific advisory committee in our DBT Northeast Center for Agricultural Biotechnology. Recently, his insect-resistant cowpea, especially the pod borer-resistant cowpea, has been deregulated for Africa, and this has been, released commercially in Africa. So I think he will be mainly focusing his talk on that particular aspect. So once again, welcoming you all, as well as welcoming TJ. I would like to hand over to TJ now, the seminar. Thank you very much, TJ.
Thank you, Bijit. I'll just share my screen now. I hope. Well, thank you, Bijit. It's a great pleasure for me to talk to my, can you hear me? Is it okay?
Yes.
Is it coming through?
Yes. Yep. Yes. Very nicely.
It's a great pleasure. It's a great pleasure for me to be here to talk to my colleagues and friends in India. I'm going to talk to you about a project that is not about India in this particular case, although I do have a long-standing interest in a similar sort of project in India with Bijit and Sumita and their colleagues in Assam Agricultural University. I'm going to talk about smallholder farmers, as most of my work is aimed at smallholder farmers. This is a typical dwelling of smallholders in West Africa. And there are many smallholder farmers in the world. 570 million is the estimate. And certainly in Nigeria alone, there's probably 30 to 40 million smallholder farmers. They're generally living close to poverty. Their income is very low, probably less than $2 per day. So this is the reason why I am interested in using some Western type technology that may be of use to them. In this project, a project like this does actually require a lot of inputs. Sorry, next slide. I want to get my next slide. Yeah, it's very important to have good collaborators in projects like this, and there are many of them, and I'd like to acknowledge them. It's also important, of course, to acknowledge funders in a project like this as well. So I'll start off by acknowledging plant breeders, the cowpea plant breeders in West Africa, Mohamed Ishiaku in Nigeria, Joseph Batieno in Burkina Faso, and Mamouni Abdullah in Ghana. These play a very important role in this project. I'd also like to acknowledge colleague Larry Murdoch at Purdue University, who was instrumental in getting me involved in this project. He's a cowpea, he's a great fan of cowpeas and has done a lot of work himself. He's an entomologist at Purdue. And I'd like to acknowledge my CSIRO agriculture colleagues as well. Finally, the funding. The Rockefeller Foundation, USAID, and CSIRO have all funded this work. It has been extremely important to have it come to fruition. And I'd also like to finish off by acknowledging the African Agricultural Technology Foundation based in Nairobi, and Monsanto for access to the BT gene that I've used in this project so far. This has been provided by Monsanto on a royalty-free basis for use in Africa. So now the crops that I'm interested in, I have worked for most of my life on the pulses, and some of them are mentioned here. Cowpea, of course, I'll talk about in a bit more detail in a minute, but I also work on chickpea with Bijit and his colleagues in India, but I've also worked on peas for a very long time and beans and some of the other pulses as well. So these are important crops because they're high protein and they have a very good quality starch. It's a low glycemic index starch. So they're very important in the diet. Global production is about 75 million metric tons, which is very small compared to the cereals, but still quite significant, as you can imagine. One of the other reasons that they're important and that I like working on them is that they fix nitrogen, and there is also a high feed value of their stubble. After the grain is harvested, the stubble is often fed to the livestock of these very poor farmers. But despite all these positives, pests and diseases are still major problems for basically all of these pulse crops. Now, cowpea is a crop of smallholder farmers in Africa, particularly, especially in West Africa. And this is the kind of farming that they do. It's not as sophisticated as some of the farming that you might be familiar with. Cowpeas are barely visible in this picture because they're intercropped with tall crops like sorghum. They grow up on the sorghum plant, but they greatly complement sorghum. They greatly complement sorghum by providing protein for this high carbon carbohydrate crop. I'd like to introduce you to this farmer. This is a Nigerian farmer, Mamodo Hahiba, and you can read what he's saying here, that he's a farmer and he sees cowpea as a main crop for him. But one of the challenges that he faces is a particular insect pest called a pod borer. And he tries to control them by spraying up to 7 times in a season. This pod borer is a serious pest. I'll give you a little bit of background information on it. The moth, it's a moth, lays its eggs on the young flower buds, and the larvae begins growth in the flowers, and then the larvae develop more fully on the pods and within the seed and in the seed. And yield losses can often exceed 50%. These are some surviving pods on a plant, but you can see that there is a lot of damage to those pods, and there's usually very little seed yield. Insecticides are used in West Africa now. They are imported usually from China without very much information on the insecticides. And spraying with them is very informal and can be quite dangerous. The insecticides are frequently moved into soft drink containers and sold to farmers. Farmers can, and there's very little information provided for them, as you might imagine. So it can be very dangerous, and in fact there are many deaths associated with improper use of these insecticides. So insect control options that farmers have are fairly limited. One option, of course, is not to control the insects at all and to rely on the seed's natural resistance. But there is very little resistance, little or no resistance in the germplasm. There's a huge collection of cowpea germplasm at IITA in Ibadan in Nigeria that has been screened, and there's basically no natural resistance to this insect pest. So if they don't control at all, then the yield is about 350 kilos per hectare, very low yield. If they use chemical insecticides, the yield can be greatly increased. As you can see there, it can be increased by 5 or 6-fold. But there is a negative side to using the insecticides. So the option that we wanted to employ was to use biotechnology to introduce inbuilt protection, which we believe would be much better. So we set about a biotechnology project we don't know, of course, in advance what the yield is going to be from these biotechnology crops. So we were hoping to increase yield by 20 to 30 to 40%, if possible. But we don't know until this has been released to farmers what that might be. So I'll just briefly introduce you to some of the technical aspects of cowpea gene technology. It's a typical gene technology project. We need a gene transfer system for cowpea, which was not available when we started. We need to reconstruct genes so that they work in the plant, so they're reconstructed into a plant format. Then we need to transfer those genes into the cowpea using the gene transfer system mentioned above. We then need to do molecular analyses of the transgenic lines, which we do, and then we do field trials to select the best lines, which might then be bred into the construction of new varieties. So that's a 6-fold step there that we go through in setting up the gene technology project for the cowpeas. The genetic transformation system that we use for cowpea is based on Agrobacterium tumefaciens, very typical, used in many other crops. We use a DNA plasmid vector for gene delivery, a binary vector, which I'll show you in a minute. And we use antibiotic selection, NPT, neomycin phosphotransferase gene, together with the antibiotic geneticin, which is in the kanamycin family of antibiotics. And the explants that we use are derived from the embryo axes of imbibed mature seeds, the cotyledonary axes that are attached to those axes. The transformation system is, I won't spend much time on it except to say that it's a typical system in which we start out with mature seed, imbibe them overnight, split the seeds apart in the morning, and then incubate them with Agrobacterium. And transfer them to agar, let them co-cultivate. And over a period of time, it takes about 6 months to go through this whole process. We induce shoots directly that start to look like plants after a few months. These shoots can be rooted in tissue culture, transferred to soil, and then to an ICU effectively, and then to the glass house where they flower and produce seed. This is the binary vector that we use to transform. It's a typical Agrobacterium binary vector with a left and right border, left LB and right border, with 2 genes, one the neomycin phosphotransferase gene, which has been converted so that it will work in plants with a promoter and a 3' end, and then the Bt gene here, which is Cry1AB, with a small subunit promoter derived from Agrobacterium, from Arabidopsis, and a 3' end, also from a small subunit gene. So effectively, this Bt gene has been converted into a plant gene, so that it will now work in a plant. This is what we have transferred into cowpea. We screen the transgenics in the lab and in the glasshouse and in the insectary. We go through a series of screenings, looking particularly, of course, for good levels of expression of the gene, the Bt gene, especially in the flowers and young pods. We also look for single copies, so this will make it easier because this is a dominant gene. It's easier for the plant breeders to use. We've got a single copy of the transgene there. From a regulatory point of view, there cannot be any vector backbone sequences there. Of course, the lines have to be made homozygous. And we look for vigorous phenotypes that look like the starting material. And we then do laboratory bioassays with the pod borer larvae, which we have in the lab. This pod borer is called Maruca vitrata. I just realized I haven't mentioned its name so far. It's not Helicoverpa, which is a major pest for chickpea. This is a different lepidopteran pest called Maruca vitrata. When all of these things have been satisfied, we then set about doing field trials in Nigeria, Burkina Faso, and in which I hope you can see there, in Nigeria, Burkina Faso, and in Ghana. These are major cowpea producing areas in West Africa. This is a typical layout of a field trial here, showing you that we are testing 6 different transgenic lines shown in color up above there and comparing them to the non-transgenic parent line IT86D shown here. Here's 6 lines. These field trials are replicated and randomized. Everything is replicated 3 times. There are buffer zones around the trial. A typical field trial, really. They're subject to the biosafety laws in each of those countries. Each country has biosafety laws. We couldn't do any trials there if there were no biosafety laws. And these trial sites are monitored 24/7. They're guarded and treated just like field trial sites elsewhere around the world. One of the first things that we had to do was to convince the plant breeders that there was no yield drag associated with the transgene in the field. This, of course, can often happen, that with resistance genes can often cause yield to drop. But one of the first things we did was to look at some of our best lines in the field in the absence of any insect and show that seed yield was no different to the non-transgenic. This is the non-transgenic parent line, IT86D, which I mentioned earlier. And here are 2 of our best lines that we thought were likely to be elite events, 709 and 252D. And this is measuring seed yield here on this y-axis, seed yield in grams per plant. And you can see that there is no yield drag associated with the transgene, a very important consideration for the plant breeders. This is a picture of the cowpeas now, the transgenic cowpeas in the field. On the right-hand side is a plot with the non-transgenic parent line, IT86D, as you can see there. And this is one of the transgenic lines on the left-hand side. And I hope you can see that there are many more pods on these plants than there are on these plants over here. We do these trials with artificial infestation, where we add very large numbers of larvae onto the plants. About 500 larvae are added onto each plant over a period of about 25 days during the flowering phase. So these plants get tested very thoroughly. This is seed yield now in the presence of insects. Again, looking at seed yield in grams per plant on this side. The IT86 shown here, the non-transgenic plant, yielding about 4 grams of seeds per plant. 709 is about 14 grams, and 252D is about 18 grams. So that's in a field trial in Nigeria. A similar field trial in Burkina Faso was like this. The insect pressure here was much higher. There was hardly any seed yield from the non-transgenic parent, but the 709 and 252D are doing very well. As you can see there, the seed yield is about 25 grams per plant. So I'm just taking a picture here to show you this is the seed, all the seeds taken off a single plant for IT86, shown on the left-hand side here, and from 709 and 252D shown over here on the right-hand side. This was in field trials, the 5th set of field trials that we had done in Africa over a period between 2009 and 2015 or so. This is a picture now showing you Muhammad Ishiaku. This is the national breeder, the national cowpea breeder in Nigeria. And what he's doing here is looking at 709A and deciding whether or not it's likely to be a suitable parent in his breeding program. The idea here, of course, is that he would cross this 709 line with some of his advanced breeding material. And that's what he has done. And he has now made varieties. This is an example of a variety that Muhammad has made, with pods shown here on the right-hand side on the transgenic, and a null line that doesn't have the transgene shown over here on the left-hand side. These are grown at the same time in adjoining plots, and you can see that this plant, even though it's the same age as these ones over here, the plants are still green because they still have lots of protein in their leaves, but nowhere for it to go, or very few pods, as you can see over here, but lots of pods on the variety, on the transgenic variety, as we had hoped and as we had expected. This is some quantitative data now from a varietal efficacy trial. These trials have been done at multiple locations in 3 different countries now over several years. And the important column to look at here is seed weight here, grams per plant, looking now at the variety rather than the transgenic line. The transgenic variety is giving about over 30 grams per plant, whereas the non-transgenic, the null, corresponding variety is only giving about 5 grams per plant. So these plants are performing very well in the field. An important feature of all of this, of course, is that we need to be able to show this to the farmers. And this is a field day where the farmers are getting an opportunity to come inside and look at the field, at the plots, to see how the plants are performing. This sort of information has been distributed to people, farmers and consumers over several years by my colleagues in Africa. So people are aware that this crop is potentially coming to them, and they take a great deal of interest in looking at the plants, making sure that they look the same as normal cowpeas do to them, despite other kind of information that they have been exposed to in the press to say that these plants may well be very different to a normal plant. So there's a great deal of interest in looking at these plants by the farmers, and the farmers get involved. In fact, the breeders involve them in selecting the best varieties for release. And this is a small group of farmers out of 20 farmers who are involved in breeder participation in selecting what varieties should be released. And we applied for approval for release in Nigeria in 2019, early in 2018, and we got permission in 2019 from the National Biosafety Management Agency. That's the regulator. So the BT cowpea has been deregulated in Nigeria so far, and They even have come up with a logo for this pod borer resistant cowpea. The variety, one of the first variety was registered in December 2019, and the seed is now being bulked up for release to the farmers. The foundation, there is breeder seed, foundation seed, and seed multiplication going on at the moment in Nigeria. We are doing the same sort of thing in Burkina Faso and Ghana, but these are a little bit further behind, probably a year or so behind in the approval process. But we plan to release varieties in those countries as well. So concluding then, what have I told you about today? Well, I've told you that we have selected one cowpea line. I should say that we have made about 800 transgenic lines, 800 independent events, and have sent 31 of those lines to the field for field testing. And out of those 31, we have selected one that as a parent in the breeding program. It gives very good protection against the pod borer and yields as well as the parent line in the absence of insects. And as I said, that's an extremely important feature for the breeders as well. So it has been used as an elite parent for breeding, and varieties have been developed in Nigeria, and they are continuing in Burkina Faso and Ghana this year. There are still, of course, I should say, we have, of course, completed the biosafety studies. They were an essential component in all of this for the deregulation. They have been completed for all three countries. Approval has been obtained, but there are still issues to be resolved. Community acceptance, as you know, as you would be very familiar with, there is still a very vocal group who are opposed to GM crops. They're very active. They're active in Africa as well as in India and elsewhere around the world. So this is a very big issue, and we have done a lot of work to inform the community about this. But it is going to be up to them in the end. We cannot force them to grow to accept this seed. But everything that we have heard so far from the farmers and the consumers, most, of course, in this case, the farmers are usually the consumers, although there are lots of urban people, of course, in countries like Nigeria. In Lagos is a huge city. So there are city consumers as well as the smallholder farmers themselves. So it is important that everybody feel comfortable about using this, and we have done a lot of work. I have not been directly involved in this usually, but we have done a lot of work informing the community about this and the fact that it is safe. We still have to get varietal approvals, of course, in Burkina Faso and Ghana. That's very close to happening. We also have a stewardship and seed system for dissemination to the farmers. This is, again, my involvement in this is fairly minor, but I have colleagues in these countries who are very much engaged, of course, in the stewardship and the development of a seed system. The seed will be provided to the farmers at a minimal cost, but it will not be free. We initially had anticipated that the seed would be given away free like other cowpea seed, but in the end, the African Agricultural Technology Foundation decided that it was essential that a seed company be involved in order to provide quality seed when farmers want to sow new seed. Farmers often save the seed for 2 or 3 years, and then they go back and get new seed. But in this case, they have decided that they want to use a seed company to provide that seed. So for us here in the lab, what we are now doing is transforming with a second gene to reduce the chance of pod borers developing resistance. and we are at a fairly advanced stage in adding the second gene, stacking the second gene to the first one, but that will take another couple of years. So I'd like to finish there by thanking you for this opportunity to talk to you in this webinar today, and I hope that you found it interesting. Thank you very much.
Okay, thank you, TJ. It's quite informative. A lot of participants have already expressed in the chat box. A very informative session. Now we are expecting questions from the participants. Let me start with oh yes, Dr. Katagiri, please.
Dr. Katagiri, yeah.
Yes.
Hello, Dr. Higgins.
Hello.
Very good evening. Is it audible? Okay.
It is evening.
Yeah.
It's dark. It's dark here. It's winter time also. It's
winter time, yes. I can understand. So the very interestingly, the number of events, what is raised is quite a good number of events are raised, 800 events. That's good.
Yes.
Only
you
one
have
question
to make
is,
a lot of events.
Yeah. To get this 800 events, how many actually number of seeds are soaked and co-cultivated? The total.
Well, about 1,000 times that, about 800,000.
800,000. Yeah.
Yeah.
That's
So it's a very inefficient system.
Very inefficient system. And the second question is, say when you soak them, and then after soaking, when you go for co-cultivation immediately, I just want to know how logically the agrobacterium really sends the tDNA meristematic tissue. Basically, it has to go into meristematic tissue. If it is not going into the meristematic tissue, it is becoming a lot of chimera. So.
Yes, yes, I understand your question.
Yeah.
The tissue that we are targeting is the node between the axis and the cotyledon. And there are lots, as you say, there are lots of meristems there, but those meristems we remove almost immediately, within the first couple of weeks, we remove all of those meristems and wait for new meristems. Some of which we hope, of course, will contain the gene, but a very few of them do, as you have just alluded to, because of the inefficiency of the system. We recover very few, but we don't actually end up with any chimeras. This system, we do in some of the other legumes that we have worked on, we do encounter chimeras, but in cowpea, we have been very fortunate that our system we do not see any chimeras at all, basically.
Wonderful, wonderful. It's my only last question, if permits, Dr. Vidyut permits. Shall I go for one more question, sir?
Yes, why not?
Yeah, that's my last question. Probably these are 800 events. You have chosen only one. I know the base for choosing one best is always good. But what is the status of all these 800 events? How were they looking like?
Well, they vary quite a lot. Many of them, of course, have more than one copy of the transgene, so they get discarded. That's one of our screening. So we don't go forward with anything that has more than one copy. Many of them, about half of them, contain vector backbone sequences. So
I see.
Many of them. So we're down to 400 already before we start looking at other issues, looking for single copy. So, and sometimes if the protein is overexpressed, if there is too much Bt protein, the plants are stunted.
Okay.
So there is yield drag with overexpression. So some of those lines get so you're down to quite a small number quite quickly. We went from 800 to about 30 before we were prepared to send very much to the field, anything to the field.
Okay.
They took 31 lines out of these 800.
Out of 800.
Yes. And then out of that, only 2 lines now.
That's really wonderful, Dr. TJ. Yeah. And I will personally I will be in chat with you with the email for further queries and and to get a lot of knowledge out of your experience. Thank you very much for this.
You're welcome.
Yeah, TJ, just I had a small query that in the field test, you got about, 6 times more yield in transgenics, 30 gram per plant,
Yep.
than in the control, it is 5 gram per plant. So what was the insect infestation percentage, in that field trial, specific field trial?
Well, these were artificially infested. So we
Okay.
put about 500 larvae onto each plant, spread out over, of course, about 25 days. But this was a huge job, a huge amount of work by my
Yeah,
entomology colleagues in Africa.
so that
They
is
are,
allowed, right? Artificially,
well,
artificial
that was
infestation
our,
is allowed.
yeah, that was our, we needed to, well, I felt we needed to do that to show that the gene expression was sufficiently high. This is much higher infestation than would probably naturally occur in
That's right,
most seasons. But
Yeah.
we went to very high levels of infestation, really, to test the gene.
Yes.
So now we don't expect 5 or 6 fold increase in yield in the field with normal infestation, because infestations is often much less than that.
Mm-hmm.
So we don't expect, but we would be expecting maybe 25% increase in yield. And because there are other insects as well, this is only one of several other insects that attack the plants in the field.
Okay, there is a question in the question answer box, one by Dr. Priya. She is asking, what might be the reasons for overexpression? As you have mentioned about some of your lines got overexpression of the gene. So she wants to know, yes,
Yeah.
reasons for
So
overexpression.
the reasons for overexpression is we used a very strong promoter. The small subunit promoter from Arabidopsis does give very high levels of expression in green tissues, and we wanted that. We wanted to have high expression, especially in flowers. So amongst those 800 lines that we made, many of them did express the BT gene at a very high level and cause stunting. So we selected lines that gave reasonable levels of expression, but no stunting.
But you can tell,
So the reason,
her, yeah, you can tell her the optimum level of expression for high level of resistance. What is the optimum level of expression?
So the level that we finally chose, the line that we finally chose, had about 5 parts per million of BT.
Mm-hmm.
And that's about 25-fold more than was needed for complete protection. So we still have a lot of protection there, 25-fold more than is needed to kill the insects. But we wanted to have that.
Okay. One more
there is
question.
another question about raising transgenic cowpea in India. Sorry,
Yes,
I
yes. Okay. Yeah, you can see that. Yeah, yourself. Yes.
Yes, I can see that one.
Hmm.
Yep. And cowpea is grown in India. It's not as important, of course, as some of the other pulses like chickpea or pigeon pea, black gram, but it is also grown in India. And it may be possible, if there is an interest in growing a GM pulse in India, to grow transgenic cowpeas there too. But we would prefer if we could get a BT chickpea released in India. We think that would be much more valuable for India than a cowpea as a first step.
Yes,
But
yes,
theoretically it's possible. Theoretically it's possible, but we do need to have permission in India to release a GM pulse, which is not possible at the moment. And we would prefer if it was chickpea, I have to say myself. And I think Bijut might agree with that also.
Yes, yes, of course.
Good evening, Dr. Higgins. Nice to see you in your own.
I can recognize Modi's voice.
Yes.
Visible too. Yeah. I say nice to see you in your own environment. We are always we see you in India.
Yep. Which I like very much.
Okay. Thank you very much. I would like to continue with your answer. You said that there are several insects and portbearer is one of them, which might be the major insect at the moment. Would you understand my question, sir?
Yes, yep. I think I know where you're going. Yes.
So once we control the portbearer through the BT, what are the likely chances of other insects become dominant and again win the crop?
Well, there are already fairly dominant insects there as well. But yes, I think I understand your question that this may allow other insects to, really now become much more damaging. And they do. They have pod-sucking bugs, and they have thrips, and even in storage, there is the cowpea. Cowpea weevil is a major problem even after harvest. So there are lots of other insects. So even though I am old, there is still lots of work for me to do into the future to help control some of those other insects. What the farmers plan to do now is, instead of spraying, they spray at the moment about 7 to 10 times per season. Many of those sprays are for the pod borer, but some of them are for the pod-sucking bugs and the thrips, especially. So what they are planning to do is to spray twice per season and not 7 to 10 times.
So
they will spray with insecticides to control the other insects, the pod-sucking bugs and the thrips especially. And I'm quite happy about this, because that to me, that is another kind of control, for another mechanism of control for the pod borer. So I'm quite pleased that they will spray. I'm not totally, philosophically opposed to chemical insecticides. I'd like to reduce them as much as possible, but if they can go from 7 to 2, I'd be very happy. And that's how they plan to deal with the issue that you're raising, which is a very good one.
Yeah.
Are you happy?
Yeah, thank you. Thank you.
There is a question.
Sir, can I ask a question, please?
Yeah, Sampa,
please go ahead.
Yeah.
Is that
Dr.
Sampa?
Professor TJ,
hello,
can
Sampa.
you hear me? Yeah.
Yes, I can see you as well.
Yeah, very good evening. You presented very nicely and very lucid manner, so it's very clear to all of us. I have a question. In chickpea and pigeon pea, normally for pod borer, people use Cry1AC and Cry2AA. But here I see that you have used Cry1AB. Is there any specific reason for that?
Yes, there is a good reason for it. 1AB is more effective against Maruca.
Okay.
1AC is very effective against Helicoverpa, but it's not all that effective against Maruca. It's probably 5 times less effective.
Oh.
1AC.
So that's why, 1AC is 5 times less effective than 1AB. That's why we chose 1AB. They're very closely related, as you know, anyway, but it's much more effective. That's why we chose it.
Okay, thank you. Thank you very much.
Can I ask one question? It's Manoj from New Delhi.
Hello, Manoj.
Yes, Manoj.
Yeah, excellent lecture, TJ. I have one specific question regarding the transgenic line. You told that has been introduced in the breeding, one of the male parent for the breeding thing. What is the status of that population? In what stage it is?
They now have 4 varieties that they have developed, 4 different varieties, and they're at the stage of bulking up their foundation seed for one of those varieties. Sorry, not for all of them. They're proceeding with the most popular variety in Nigeria. They use that as the female parent in the first instance, just so that farmers would feel comfortable about using a new variety with the BT gene. It's basically the same as the line that they're very happy with, that they use a lot, and it's easy for the breeders to introduce a GM version, which looks like, you know, basically the same.
Okay. And what about the other three lines? Other three varieties that you talked about, that one you have told that they're the most popular one. They
Most advanced, but they have breeder seed and foundation seed, I think, of the other three. But they're not pushing ahead with those this year. Farmers will get the seed this year, so
Okay.
small amounts of seed this year, and they'll get it with this first Sampia 20, which is a derivative of Sampia. They're the most popular variety in northern Nigeria.
Is there any biosafety issue regarding this breeder seed?
No, they have been approved by the regulator and they have been approved by the Varietal Registration Authority.
Yeah.
Did you have, was that?
So the same thing can be done in India also. Then I think there is no problem.
Well, similar, yes, India has similar biosafety authorities, but at the moment, I think it's not possible to release a GM pulse in India.
No, I'm not talking about the GM pulse, because if you are using the breeding lines to improve the resistance in case of all the release varieties. For breeding, I think that if it comes once in the breeding thing, then I think the biosafety issue is
even then, Manoj, even then, because this is going on. Our chickpea lines are being used in US Dharwar and then also in Punjab Agricultural University. They are doing the breeding now using our lines developed in collaboration with Australia, and then they are already going ahead. It's, I think, now about BC4 generations. But yes, even that for releasing those as a variety in the field, you have to deregulate them, because the original line is transgenic one.
No, that's
yes,
fine.
because
Yeah, Manoj, please.
For the gene editing also, for the gene editing line also, we have to do the same way. That we have
for
to
gene editing, our last seminar, for our last seminar was on genome engineering, and she basically, Dr. Bibha Ahuja, who is one of the members in the genome editing, regulations here in India, and then they are going to recommend very soon, what kind of recommendations will come, we'll see. But yes, there is a committee formed by the government of India for genome edited lines. What will be the strategy for deregulating them? So this is going on, and for the transgenics, whether they'll be treated as transgenics or differently genome edited. But yes, these things will come soon.
So still to be sorted out. But
yes,
I'd like to just make clear that even though conventional breeding has been used to make a new variety, it still has the transgene in it. They took the transgene out of the line 709 and transferred it across into Sampia. So it's still transgenic as far as the regulator is concerned.
Yes.
So in case I didn't make that clear, Manoj.
Okay.
Could I ask?
Okay.
Could I answer another question from Manab?
No, I think Nagendra Singh is waving his hand.
Okay.
NK Singh.
NK Singh,
okay.
Yes.
Good evening, TJ.
Hello, Nagendra. Good to see you.
Yeah, good to see you too, and very nice talk. I thoroughly enjoyed it. I have three questions. The first one is that you said that so many of your lines has a lot of vector backbone.
Yep.
What do you screen for the vector backbone, and what method do you use for that? That is the first question. And the second one is that you said that there is a lot of opposition to GM crops in Nigeria also. So how do you handle that issue at the practical level? And the third one is that
you have three questions. You're already asking me two difficult ones. Well, no, one very difficult one and one easy one, of course. But go ahead with your third one.
Regulatory. And the third one is the related one. What is the status of regulation of genome edited plants in Australia?
Okay. So going back to your first question, we use southern blotting to look for backbone. So that's what we've mostly done up till now. But now we are using genome sequencing to look for any additional DNA, because the genome of cowpea has been finally published, well, fairly recently by Tim Close and his colleagues in Riverside. We can now sequence the transgene, the genome of the transgenic lines. So we're starting to do that, but we do start out with southerns as a first step for looking for vector backbone sequences.
So how many, TJ, how many out of 800 you analyze for the vector backbone? I mean, what is the first filtering?
We probably did maybe 200.
Okay.
Because, you know, we discard quite a lot of lines because they have multiple copies.
Okay.
We find that about half the lines have multiple copies. 40, maybe 40% have single copies. So that brings the number down quite quickly. And if we have too high level of expression, we don't proceed with those ones. So we didn't have to do southerns on all 800, because I think you know probably as well that southerns are not all that easy, even on genomes that are fairly small compared to wheat. This is only a 620 megabase genome. It's still not easy. Your second question was, what was it?
Opposition in Nigeria up to this GM technology. How do you handle?
Oh yes,
yeah.
Opposition. There's a lot of opposition. Friends of the Earth are very active there. There are many groups that come from Europe, mostly from France, particularly, that they are highly subsidized by the European people in the European Union, especially, and they're very active. So they have taken the regulator to court several times to try to stop them from approving the BT cowpea. But each time the court has ruled against them. But of course they will keep coming back.
Yes.
And your final question was, what is the status of gene editing in Australia? Well, gene editing, I think that single base changes will not be regarded as GM. It will be treated in the same way as mutagenesis.
Okay.
So SDN1 will be non-transgenic. SDN2 and 3 will be treated as transgenic by the regulator here.
Okay.
Okay, great. Thanks, TJ. Thanks. Very good.
Thank you, Dr. Singh. Now, question from Manab.
Manab, yes, I can see the question from Manab.
Yeah.
It's and it's related to the question that Nagendra was asking about
yeah, same.
Yeah, that how do you convince the farmers about GM cowpea? Well, I think the farmers, as far as we can see, this is all done. These are surveys basically done by social scientists. The farmers do not want to use chemical insecticides if they can possibly avoid it. So I think they are relatively comfortable about GM cowpea. But of course, they're continually bombarded by Friends of the Earth and, to a lesser extent, Greenpeace and other civil society organizations in Africa. They're just as active there as they are in India and elsewhere around the world. So I think that, we are still waiting. I don't know for sure that the farmers will accept them, but everything we have been told by all the surveys by the economists and the social scientists, they say that farmers are very keen to have them. They don't want to be spraying so much with chemical insecticides.
Yes. There's a question from Ruby Gupta. What might be the reason that the lines expressing very high level of BT show stunted growth?
Yes, I don't know. I'm sorry, Ruby. I don't know the answer to that. I'm assuming that the protein is binding to some receptor in the plant as well, and is actually causing some detrimental aspect to the growth. But we have not followed up on this. I don't know the answer. But in some of the work that I have done with Bijit and Sumita,
yes,
we have observed similar things in chickpea. And there are also some publications in rice that also say that too high level of expression of BT can cause stunting
Yes.
in cereals as well as in pulses.
Mm-hmm.
So I'm sorry, I don't know the answer to that question. I'd love to know the answer to it, but we don't know it. So there's a question from Jayanta.
Yes.
Is there any environmental factors increase of pod borer? I don't understand that question. Can you tell me what is meant there?
Jayanto, sir, is there any environmental factors? Means whether insect infestation may be enhanced because of certain environmental factors. Whether environment like, you know, temperature or maybe humidity. Yes, these things sometimes, you know, do with the insect infestations, you know.
Yes, there are many factors, many environmental factors that affect the level of infestation every year. You know, some years infestation is 80% of the crop is lost. Other years it's only 20%, and this is, you know, most likely due to different environmental factors that we don't know about. And of course, we also are going to be faced with climate change. We are already facing climate change, and this may have an effect as well. Might be a negative effect, but quite possibly it will be a positive effect as well on these insects. We don't know that yet.
Yes. There's a question from Dr. Patil. MD Patil.
Okay, I see. Did you conduct any study on bruchid infestation on the transgenic versus the non-transgenic seeds? Oh yes, this is a good question, interesting question. Bruchids, Colossa bruchis maculatus is a very severe storage grain pest, and huge amounts of the crop are lost during storage. We have tested the BT gene, and it has no effect on the bruchid at all. We have looked at another BT, a Cry3, and also it has very little effect on the bruchid. We do have a gene, which we have also tested in chickpea with Bridget and Sumita, an alpha-amylase inhibitor from beans, Phaseolus vulgaris, and that is very effective. But we haven't yet gone through the deregulation of that gene. But we have made transgenic cowpeas with the alpha-amylase inhibitor gene, and it does give complete protection against the bruchid. But we haven't taken that through all the processes that I've talked about today. But we would like to do that in the future because it is such a serious pest.
Yes.
Hello?
So BT obviously will not work, but other genes like
BT
alpha
doesn't work.
Yes. Alpha amylase inhibitor from common bean will work against bruchids.
Yeah. There is a question from Alok. He has raised his hand there.
Yeah.
Hello, Alok.
Oh.
Hello, sir. Hello, TJ. Yeah, I am Alok from Indian Institute of Pulses Research, Kanpur.
Yeah, he knows you.
Yeah.
I know who you are.
Yeah.
Yes, of course.
Yeah. Thank you very much for your presentation. I had a two question. Did you observe some variation in the expression pattern once the plant moved from vegetative stage to the reproductive stage? And my second question. And the second question is, have you identified the site of integration of the transgene? And which strategy, if yes, which strategy have you used?
Okay, so the first question, yes, was something that we did very early on, which was to look at what happens to expression of the gene as the plant ages, as it goes through development. And the protein is degraded at the same rate that other proteins are. If you look at, say, the oldest leaf on the plant versus the youngest leaf, there's about a 5-fold difference in the level of total protein, and there is about a 5-fold level decrease in the level of the BT protein. So it follows total protein pretty much. It isn't more stable, for instance, or it's very slightly more stable. But we're not all that interested in expression in the leaves. We're much more interested in expression in the flowers and in the young pods and in the seed. And expression is quite high in the flowers. you wouldn't naturally think that with the small subunit because there are very few plastids in the flowers, but in fact there is quite high levels of expression in the flowers and in the young green pods and even in the young green seeds. So that issue wasn't wasn't a major issue for us because the insect doesn't really attack the leaves or petioles unless there is a huge, very, very high level of infestation. But even if there was, there is still sufficient BT there in old leaves to give complete protection, even though the protein level does drop with other proteins. I'm sorry, I've forgotten your second question. Oh, what site of integration, I think you asked about. Yes, we do know the precise site of integration, and we did that by sequencing the genome. We did whole genome sequencing, comparing it to the non-transgenic line. It's now, cost effective to do sequencing of the whole genome, and because the genome is sequenced, we can.
Which platform did you use, TJ? Yeah, sorry, yeah.
So we used nanopore.
Nanopore, okay.
Yeah, it's very good for very large pieces of DNA. Okay. We do then, additional sequencing once we've got. There's actually, quite a few errors with nanopore, but then as soon as we had the sequence, we were able to then make event-specific markers and sequence for a couple of kb on either side of where the gene has gone in. Yeah. So that's our preferred way of going now.
Thank you, TJ.
Thank you very much. There was another question from a veterinary microbiologist working on
the same person who asked the earlier question of high expression, region for the high expression.
Yeah. Yes, I see your problem is weak expression. You need to test more promoters. We have looked at other promoters as well and have found some, many that were too weak. But the small subunit, the one that I mentioned in some detail, that one is a very high, is a very strong promoter. But so, if it's possible, please test other promoters. Would be my advice. Have we run out of questions?
Is there any more question? Attendees? Any more questions from participants?
I don't see any on the question and answer. There might be other questions that people have.
In the chat, I am checking. No, they are all, thankful to you for nice sessions only.
Okay.
Yeah.
Hello.
Oh, somebody is asking question. Yeah.
Yeah, this is Surendra Paul from NBAIM, National Bureau of Agriculture Imported Microbes.
Hello.
Hello, TJ. I'm Surendra Paul from National Bureau of Agriculture Imported Microbes.
Hello, Surendra.
Okay. Yeah.
Please go ahead with your question.
Yeah, my question is, with the time, insect may get resistance to the gene product which you are expressing in the plants. So they may get damaged more severely that to the transgenic plants. So what are your suggestions how we can deal with this kind of problem?
Yes, this is a problem, and it's one that we have been thinking about from the beginning. What we're doing now, of course, is to add a second gene with a different mechanism of action. So the likelihood of insects building up resistance to a second gene is much lower. So that's what we are doing now. We have a second gene that we have introduced and have done some field trials already with that second gene, and we know that it works in the field. So my colleagues in Nigeria have already started backcrossing with that new line with the second gene. So there will be two genes stacked together. So that's one of our first steps. The other one I did mention briefly, I think in answering Nagendra Singh's question, where it wasn't again, where I said that the farmers are spraying with chemical insecticides. And I'm quite happy about that, because I think that's also will help because it will kill any surviving maruca as well as the pod-sucking bugs, etc. So my preference is that we have more than one mechanism so that we reduce the chance that the insects will build up resistance.
So
I should also say that in West Africa, there are many other hosts for maruca besides cowpea. So there are many other plants that will act as a kind of refugia, if you like. There are many, for instance, legume trees, tree legumes that are also very much preferred by maruca, really. So I think there are many other hosts besides cowpea as well for them.
So you mean to say that there is no harm in using a little bit of this chemical pesticide also with this transgenic kind of plants?
Yes, well, that's my own preference, and that's what the entomologists are recommending in Africa, that they should spray 2 times a season, perhaps, depending on the level of pod-sucking bug infestation, etc. That there is a recommendation from the entomologists that they should spray, and the farmers, I think, would agree to spray 2 or 3 times. But they don't want to be spraying 7 to 10 or 12. Sometimes they're spraying 12 times in a season. Now, some of them, an average would be 7 or 8 times. But if they could reduce that to 2, this would be very much better.
Yeah, that is what we are
Thank you very
practicing
much.
for cotton as well here in India, for BT cotton. We are reducing the spray, number of spray.
Thank you very much.
There is another question I have seen. Some Dr.
Is it harmful for humans?
Yes.
Yes. Is it harmful for humans if we used as food? Well, yes. So cowpea is a food crop. It's not really a feed crop. The stubble, the straw that's left over after harvest, is fed to livestock, but the cowpea is really a food crop for farmers and consumers in Africa. So we have to feel convinced that this is not going to be harmful for humans. And I think probably the best evidence is that organic farmers have been using BT sprays on food crops for 70 years, and we have been eating it for a very, very long time. I think that's probably the best evidence to say that BT is not harmful for humans.
Yes.
There have been many, many studies on this, of course, as well. But that is a piece of practical information to say that we have been using BT sprays of this kind for a very long time.
Yeah, from regulations point of view, also you have to confirm that it doesn't lead to any extra protein digestibility or something like that. Those things need to be tested. Yes.
They have already been done. All of these tests have been done. Yeah.
Yes. So if there is no more question, Dr. Manoj is asking some question again. Dr. Manoj?
No, sir. I have already asked.
No. I have seen your name there, in my screen. So
yeah.
I thought
you're allowed to ask more questions, but
anyway, it was very yes.
Yeah, this is Dr. Katgiri again.
Yes.
This is not a question.
Yeah.
It is just a kind of how to go ahead with the transgenics in India. That's why I'm asking Dr. Higgins a kind of suggestion. Why can't you have a kind of international level kind of committee or kind of experts who can suggest all over the world that transgenic technology is absolutely not a harmful technology, and all this kind of care is taken. Why can't you do this kind of activity? From somewhere it has to start, because now in India we have a lot of events are there in in the backyard of all of us, but unfortunately nothing is coming up. What's your opinion?
Okay, Dr. Higgins' opinion, yes, first. Yeah.
Yes. Well, yes, this is a very good suggestion. I have spent the last 30 years in Australia talking to people about GM crops. We run courses for newspaper reporters, for politicians, and so on. It is very difficult to persuade people who have an ideological position that they are opposed to this. I should also say, acknowledge that 127 Nobel Prize winners.
Yeah, that's the same thing I wanted to say, yes.
They have written a letter.
Yeah.
They have signed a letter to say that GM crops are safe and should be deployed around the world. Yeah. So there's a lot of information out there, but it's not enough to convince some people who are very effective advocates for their position. They're probably a very small proportion of the total population, but they are politically very, very active.
Yes. Very, very active. Especially for the golden rice, they say this should be released very soon. Yeah.
Yeah. So, thank you.
Yeah.
So, if there is no more question, TJ, do you have any special or specific comment to the or suggestion to the participants or any?
No, other than I'm very grateful for this opportunity to talk to my friends in India, whom I like very much. It's always a great opportunity. I always value an opportunity like this to be able to talk to people. So that's all I wanted to say. I wanted to say thank you very much for this webinar.
So TJ,
oh, somebody's asking a question again. Yes.
Manoj again. Manoj.
Oh, okay. Manoj,
go ahead.
Please see that in the chat box. I just send a link for Professor Pental's article on BT cotton. That was the Kranti has written in Nature Plant and a commentary, and then in a rebuttal, Professor Pental has commented. So, so you please go through this rebuttal later by Professor Pental.
Yeah, I think TJ has already seen that because we sent him.
I may not have seen that most recent one. I've seen a rebuttal.
Oh, earlier one, yes. Maybe this is a recent one.
This is a recent one. This is
a recent one.
No, but this is the recent one, man.
Okay.
Please go through it.
Okay.
I saw the paper, the original paper by Kranti. I was quite surprised by it.
Yeah, and Professor Pental was rebutted and excellently written by Professor Pental. If you just go through it, you will be loved it to read it.
Okay, it's very important that DP Pental do that as well.
Yeah,
it's very helpful.
Yeah. Dr. Pental sir has been trying a lot for his mustard in India.
Yes,
but still, it's very difficult to convince.
I would love to see it released in India. A crop that's so important in India as well.
Yes. Yeah.
So
if there is no more question, then, once again, from the organizers' side, I would like to thank everyone. And I have seen, it was very smoothly going, today's presentation because, we shifted from Google Meet to Zoom. For that, of course, we had to spend some money, but, it's worth spending. I think because 150 participants continuously present or attended this web seminar. So once again, thank you, TJ, and love from our side, and we disturb you, of course, because it's your bedtime almost. So go and
no, it's not even dinner time in India.
So it's not dinner time. It's not dinner. Oh, in India, yes, for you. When you come to India, it's dinner time, of course. Yeah, that's right. You are true. You are correct.
Your dinner is even later. Thank you to everybody for their nice comments, for your very nice comments.
Yeah. Thank you very much.
Once again, thank you, DJ.
On 19th, we are going to meet with another, expert, Dr. Manoj Prasad. So he'll be talking on small grain, pulses, small grain millets.
Millet.
Millet,
Okay.
yes, millets,
Yep.
yes.
We look forward to hearing that.
Yeah, on 19
Yeah,
at 11 o'clock. Of course, for TJ it will be done 7 o'clock in the morning. At this seminar will be at 11 o'clock in the morning, Indian Standard Time.
Oh no, that will be 3:30 in the afternoon here for us. So that's very good.
Sorry. It will be
Yeah.
it will be 3:30. That's I was going other way around. Anyway, it will be 3:30.
Yeah.
So yes,
that's a very good time for me.
Yeah.
Yep.
yeah.
So see you all once again on 19th. Thank you very much.
Thank you.
Bye-bye.
Bye.
Thank you.
Bye.